right, I think we'll start up with Jennifer Raymond from Stanford.
All right, good morning. Yesterday, someone asked me, when is the cerebellum session? And this is it, the next 30 minutes. And I may be the only one in the room who works on the cerebellum, but hopefully some of the ideas that are coming out of our work on cerebellum will be useful to many of you in thinking about other brain areas as well. And I will echo at least one of the themes you heard yesterday in Jim Knierim's talk on the medial temporal lobe, where I'll also be telling a story whereby we're finding heterogeneity in what had previously been considered kind of a monolithic brain area. And the heterogeneity I'll talk about has to do with the properties of synaptic plasticity. So my lab essentially is interested in understanding the logic of how neural circuits tune their own performance through experience. And that logic is embedded in the rules that individual synapses use to determine whether they should change or stay the same. So an individual synapse doesn't know what's happening in the world, but is making a local decision. Do I need to change or stay the same based on the pattern of activation it experiences? So my lab is interested in better understanding those rules, and then ultimately how the rules at different synapses within a circuit are coordinated so that together they define an effective way to tune that circuit's performance. Now, of course, we have some ideas about the rules governing plasticity at synapses from the work that's been done over many years in brain slices, where we know, for example, many synapses have Hebbian plasticity, whereby cells that fire together tend to wire together. Two cells are coactive. The synapse between them tends to get stronger. We also know about spike timing dependent plasticity, whereby you can get long-term potentiation or depression depending on whether the presynaptic neuron fires before or after the postsynaptic neuron. And I think the general assumption has been that there are just a few generic forms of synaptic plasticity of these types that are implemented at synapses throughout the brain. And the way that you get specialization for a particular learning task, the assumption has been, comes not from the synaptic mechanisms, but from the way they're embedded in a particular circuit architecture. At least that had been my assumption. But what I'll show you today is that the synaptic plasticity rules themselves can be precisely tuned for a particular behavioral task. So that's going to be the take-home message. We discovered this by working on plasticity at the parallel fiber to Purkinje cell synapses. And almost exactly 50 years ago, David Marr and James Albus hypothesized that long-term depression, an associative form of long-term depression at the cerebellar parallel fiber to Purkinje cell synapses, is a key mechanism for cerebellum-dependent learning. And I think there's some truth in this idea, as I'll say a bit more about in a minute. So let me take you through this kind of textbook model of cerebellar learning. The Purkinje cells are the sole output neurons of the cerebellar cortex. So you can think of them as representing all of the different responses that the cerebellar cortex might want to make. There are two inputs to the cerebellum. One comes from mossy fibers via the granule cells, which make the parallel fibers. You can think of them as representing all of the different contexts in which you might want to make a response. So by modifying the appropriate set of parallel fiber to Purkinje cell synapses, you can learn to make the right response to a given context. The other part of this model has to do with the other input to the cerebellum, which comes from the climbing fibers. They come out of the inferior olive in the brainstem. And they seem to carry feedback about the quality of the response. And Marr had hypothesized that this feedback was a reinforcing signal that would strengthen synapses that led to a good response. Albus hypothesized that this feedback was an error signal that would weaken synapses that led to a bad response. And it was Albus's error-correcting version of this story that's best supported by the physiology. Because if you record from climbing fibers during a variety of tasks, they seem to encode errors. And in slices, climbing fiber activation can induce an associative long-term depression of the parallel fibers that are co-activated. So sometimes this is referred to as an anti-Hebbian form of plasticity because the associative form of plasticity is depression, and if you just activate the parallel fibers alone, you get potentiation. Okay, so this is the textbook model of cerebellar learning, whereby you get a gradual sculpting away of synapses that lead to errors through this associative form of long-term depression. My lab studies this form of LTD and its role in learning in the context of this behavior, the vestibulo-ocular reflex, or VOR. I'm really proud of my performance as the R at one of our lab ski trips here up in Tahoe. So this is a reflexive eye movement that tends to stabilize images on the retina. When the head turns, this movement is sensed by the vestibular system, which drives an eye movement in the opposite direction. So if the head turns left, the eyes turn right, and vice versa. So this reflex keeps the eyes pointing in the same direction as the head moves about in the world, and that improves vision by stabilizing images on the retina. In order to do its job of stabilizing images, this reflex needs to be well calibrated, and that calibration is implemented through cerebellum-dependent motor learning. If for any reason the VOR fails to stabilize images, the cerebellar learning kicks in and adjusts the amplitude or the timing of the eye movement responses until images are again stable during head motion. This kind of simple form of learning maps very nicely onto the more albus model that I described. In the relevant part of the cerebellum, which is the flocculus, the parallel fibers encode the vestibular signal that drives the response. The Purkinje cells, if you activate them electrically or optogenetically, can drive an eye movement. And the climbing fibers in this part of the cerebellum encode what I'm calling retinal slip, image motion on the retina. And the goal of this reflex is to stabilize images on the retina. So if there's image motion during head turns, that means there's an error. The climbing fibers encode that error. So there's this nice mapping. Masao Ito first pointed this out in the 70s. Nice mapping of the simple ocular motor learning task onto the classic Mar albus model of cerebellar learning. Over the years, there's been a lot of debate about is LTD the mechanism of cerebellar learning or not. Work in my own lab suggests that under particular conditions, this LTD mechanism is recruited during cerebellar learning, and slight changes in how you do the training can cause LTD to not get recruited. That's a different story, but we think that this is at least sometimes recruited to support learning. There was a question.
We are not born with this reflex. It's something like that as children we learn to do, or?
Yeah, there's a 3-neuron reflex arc through the brainstem. So you can lesion the cerebellum and you still have a VOR, but in order to calibrate it, you need the cerebellum.
So,
and in a similar way, if you lesion the cerebellum, people are not paralyzed, but their movements are not well calibrated.
so this process keeps being calibrated again and again?
Again and again. So you would tune it up during development, but during change, as the muscles change their strength during development or aging, you'll need to recalibrate again. When you get a new pair of glasses with slightly different magnification, you need to adjust the gain of your VOR. So the idea, like any movement, we initially calibrate it, but we're constantly monitoring the accuracy of our movements and adjusting it through cerebellar learning.
Where is the error signal actually computed?
So it's an image motion signal. It's coming through largely subcortical visual pathways, nucleus of the optic tract. But that's another interesting question. We usually study this, learning under conditions where the whole world moves in a way that we can control. But under normal conditions, things are moving in different directions on the retina, depending on where things are relative to my focal plane. And so that's another whole interesting question of under that condition, what image motion drives the climbing fibers that we've looked at a bit too, but that's a different story.
Question? So the cerebellum knows about the depth sensitivity of the VOR.
The cerebellum knows about the
Depth sensitivity.
yeah, so we don't know, there is, you do adjust your VOR for different depths. So you have a higher gain when you're fixating something near versus far, and those need to be independently calibrated. That's not the main point of what I'll talk about here, but it is exquisitely calibrated. The amplitude and timing of the eye movements are exquisitely calibrated, and I will show you that timing part of it, which is...
Yeah, if you get about one climbing fiber spike per second, right? Mm-hmm. Seems like insanely high learning rates. Anybody has sort of worked out if that's necessary, or what's, given how fast the VR drifts?
Yeah. So this is a question of, do you get learning every time that the climbing fiber fires? And does the climbing fiber fire every time there's an error? And I'll show you a little bit of that. So let me keep going here. And okay, so we had evidence. We had our lab has, despite controversy about does LTD contribute or not, we had in our lab recording, stimulation, and perturbation evidence, convergent evidence that at least under a particular set of conditions that we can define, this mechanism got recruited. Yet, there was this thing that had always bothered me ever since I was a postdoc with Steve Lisberger. The thing that had bothered me is how do you use a coincidence-based associative plasticity mechanism to support learning in the face of feedback delays? So this is a problem not just here, it's a problem for any kind of feedback-based learning. How do you use coincidence-based synaptic mechanisms to do that? And here, when I was a postdoc, we estimated that the delay for the climbing fibers to respond to an error is 122 milliseconds. We got that specific number fitting the phase shifts that we got across different frequencies with a constant delay. We then fit mouse data from another lab, that was monkeys. Mice, we got 121 in mice. So there's a pretty consistent delay, at least under high contrast visual conditions, for the climbing fibers to respond. A pretty long delay, and the question is, how does the system deal with that? One possibility was suggested by what had been described in the synaptic literature, which is that there's a pretty broad coincidence window had been reported for these synapses for inducing LTD. And so a lot of people had used coincident parallel fiber and climbing fiber activation. This is work from Wade Regere's lab at Harvard showing they could get the LTD when the climbing fiber was delayed 50 milliseconds or 150 milliseconds relative to the parallel fiber. And even if the climbing fiber, which supposedly has the error, was stimulated before the parallel fibers, they also got LTD. So there's a window of a couple of 100 milliseconds that might solve this delay problem by being broad enough to accommodate those long delays. However, that never quite sat right with me because one of the things we know the cerebellum does is very precise timing. And even a weekend athlete, even you and I, can time our movements with a precision on the order of a few milliseconds. So if you're throwing a ball, the difference in timing between the release and the arm movement, if you're 5 or 10 milliseconds too early, you will miss the target way too high. And if you're 5 or 10 milliseconds late, you will miss it way too low. So you and I can time our movements with a precision on the order of 10 milliseconds. You could imagine ways you might do that with super resolution in this kind of sloppy synaptic mechanism, but I'll show you that that doesn't seem to be the case. Also we had evidence in vivo that we can precisely time our eye movements. And this had come from doing a trial-by-trial analysis of plasticity in the intact circuit that modeled after what Javier Medina had done with Steve Lisberger. So Javier said, and this gets back to your question, Peter, climbing fibers have a low firing rate of about 1 hertz. And even if there's an error every trial, a given climbing fiber will fire on some trials and not fire on some trials. And so Javier hypothesized that if the climbing fiber is what's driving the plasticity in this circuit, then every time the climbing fiber fires, we should be able to detect the induction of a little bit of plasticity. And on trials where it doesn't fire, we would not see anything if it's the driver of the plasticity, and so on. And the way he did this analysis and the way we did is by using single recordings from Purkinje cells. And you can record 2 kinds of spikes in the Purkinje cell. Normal sodium spikes that are called simple spikes. They are coming out of the Purkinje cells at 50 or 100 spikes per second. And then there are these complex spikes, big calcium spikes, that are the signature of a spike in the Purkinje cell's climbing fiber input. So each Purkinje cell gets input from a single climbing fiber, and each spike in the climbing fiber produces one of these characteristic complex spikes. So with a single recording, we get 2 pieces of information. We see the error signals coming in on the climbing fiber input, and we see what happens to the Purkinje cell output. So what Javier and we did is look for pairs of trials where there's a spike in the climbing fiber on one trial, and we ask what happens to the Purkinje cell's output on the next trial. And if the climbing fiber is inducing a little bit of depression in the excitatory parallel fiber inputs, maybe we'll see a little bit of a decrease in the Purkinje cell's output on the next trial. And much to my surprise, that's what they reported in 2008. So here are their results. averaging together all of the trials where the climbing fiber fired, and then looking at the trial-to-trial change in Purkinje cell output. They saw that when the climbing fiber fired, the Purkinje cell fired less on the next trial. If there was no spike in the climbing fiber, there was no change. So there was a very tight correlation between the occurrence of error signals on the Purkinje cell's climbing fiber input and the changes that it underwent. So we did the same analysis, but with one key difference, and that is in how we aligned this analysis. Oh, and I should point out that in their work, they saw this kind of 200 millisecond window during which the Purkinje cell decreased its firing. That's very reminiscent of what I showed you in the Synaptic data as well. But we redid the analysis, and whereas they had aligned their analysis on the time of the trial start, we, a student in the lab, Hannah Payne, redid this, aligning on the time that the climbing fiber fired within the trial instead of on trial start. And then she asked, what happens to the Purkinje cell's output at that time in the next trial? And when she did this, this amazing temporal precision fell out. So what she saw is there was a decrease in Purkinje cell output on the trial after a climbing fiber spike. But that decrease didn't happen at the time when the climbing fiber fired, but at an earlier time, about 100, 150 milliseconds earlier. So it looks like the climbing fiber, when it fires, is not inducing synaptic changes in the synapses active around the time the climbing fiber reports the error, but at the earlier time when the error was getting generated, seeming to at least partially solve what theorists call the temporal credit assignment problem. Peter.
I think I just missed something. So you're basically, you have a climbing fiber spike and you're aligning on the next climbing fiber spike?
On one trial, the climbing fiber fires. So if the climbing fiber fired 200 milliseconds into the trial, then we ask what happens to the Purkinje cell output. We're looking at the trial-to-trial change in Purkinje cell output aligned on that time. So on one trial, the climbing fiber will fire at 200 milliseconds into the trial. Another time, it'll fire 300 milliseconds in. So we're aligning on the time of the climbing fiber spike. And we're saying what happens to the Purkinje cell output relative to that. And so what I'm showing here is that there's a little artifact at 0, but around the time the climbing fiber fired, we don't see any change in the Purkinje cell's output. It seems to be inducing plasticity in the synapses that were active 100 or 150 milliseconds earlier. And that's the time that the error was generated, not the time that the error got reported back in the climbing fiber. And we see it not just in the Purkinje cell's output, but we also see it in the eye velocity, which suggests that when the climbing fiber is fired, there should be some pretty good correlation across the whole population. We can talk about that. Okay, so that still left us with this disconnect between this kind of broad coincidence window that had been described for the synaptic plasticity and the kind of temporal precision on the order of tens of milliseconds that we needed to account for what we saw in the intact circuit and the behavior.
So do you mind going back one? I was just trying to understand, because at some level, I mean, it has to do with where your confidence bounds are, but the curve looks like if you did this a million times, you might see that the deviation is broader, at least there. Yeah, the red one.
Yeah, that there may be a little, there's a
Well,
trend
I'm just
here.
thinking like from negative 200 to negative 100, right? I mean, I'm just trying to figure out like what level of precision do you feel like is actually there from that, and what level is sort of a statistical inference issue?
Well, I'll show you this, what we think is the synaptic mechanism of this, and we also haven't narrowed it down to the, is it 5 milliseconds or 10 milliseconds, but it's very similar. So let me keep going so I can show you the results, and then we can talk about them together. So a very talented postdoc in the lab, Aparna Suvarthan, found, I think, what is the resolution of this apparent discrepancy between the synaptic properties and what's needed to support the behavior. Aparna just started her own lab at McGill last month. And what she found had to do with comparing different parts of the cerebellum. The properties of plasticity at the parallel fiber to Purkinje cell synapses, I'll show you, differ in different parts of the cerebellum. Previously, almost everything we knew about the synaptic physiology of the cerebellum had come from the vermis, which is on the midline, because it's easy to slice and keep everything in the plane as you like. Aparna made slices of the flocculus, which is the part that supports the oculomotor learning tasks we study in my lab. And when she did that, she found the properties of synaptic plasticity were quite different. So first she made slices of the vermis. She did the experiment everybody does, 300 coincident activations of parallel fibers and climbing fibers. She got LTD like everyone else has reported. And then she did the same experiment making slices of the flocculus, and instead of LTD, she got LTP. And I mentioned LTP is the non-associative form of plasticity at these synapses. It's what you see if you just stimulate the parallel fibers. So it's as if the slice didn't even see the climbing fiber activation. So the first lesson for us was that there's regional variation in the plasticity rules, even for the same anatomically well-defined type of synapse. The cerebellum is described as crystalline in its architecture, the same 5 types of neurons repeated in the same circuit architecture over and over throughout the whole structure. But what we found was that the functional synaptic properties differed in a dramatic way between regions.
Jennifer, so is the feedback much faster in the vermis? I mean, why?
Yeah, so let me show you the data and then I'll talk about that. So we thought about this 122 millisecond delay that the circuit would normally see between parallel fiber activation and the feedback on the climbing fibers in vivo. And when she stimulated the slice using that delay for the climbing fiber, then she got a beautiful long-term depression. So she could get LTD when she used the right delay, the behaviorally relevant delay. This is a summary of that. So coincident activation, unlike what had been reported in the vermis, gave us LTP. If we delay the climbing fiber 120 milliseconds, we got LTD. Thanks to the proverbial reviewer number 3, we repeated this in a number, did small variations on this. We measured the changes in current clamp or voltage clamp. We used a single parallel fiber stimulus or a little train. It didn't matter. We got the same answer no matter how we did it. Coincident activation, potentiation, 100 millisecond delay, depression. Tuning for that delay was quite precise. It gets back to Lauren's question. If we used a 100 millisecond delay for the climbing fibers, we got significant potentiation. If we used 150, we got nothing significant on average. So we really only see depression with that behaviorally relevant delay that the error signals would normally take in vivo. This form of LTD that we found is unlike any synaptic mechanism that we know of in other synapses. It's similar to STDP in that there's a window of a few tens of milliseconds that's effective for inducing the associative plasticity. But unlike the STDP at most synapses where that window is close to coincident, in this case it's shifted by more than 100 milliseconds from coincident. And then just to compare with what had been described in the vermis, and not just by Wade Regere's lab, but other labs as well, there'd been this broad window described. To put these on the same time scale, you can see that what we see in the flocculus is an order of magnitude more temporally precise. Okay, but we had a hard time believing that the flocculus was precise and the vermis was not precise because, as I said, we think of the cerebellum in general as doing temporal precision. So we went back to the vermis and we were able to find precision there as well. We did this by using a form of short-term plasticity. So we did a single pairing of parallel fiber and climbing fiber activation and asked how that changed the parallel fiber to Purkinje cell synapse. Analogous to that trial-to-trial, single trial analysis I showed you in vivo. And when Aparna did these experiments in the flocculus, she saw the same tuning for 120 milliseconds for the short-term depression that she had seen for the long-term depression. When she went to the vermis, she found she could get the depression with coincident activation. If she delayed the climbing fiber 100 milliseconds or 120 or 150, it was associative. If she didn't stimulate the climbing fiber, she didn't get it. We had other controls where she didn't stimulate the parallel fibers. So it required conjunctive activation of parallel fiber and climbing fiber, but it seemed to have this broad coincidence window like what had been described for LTD. However, if she then looked at individual cells, she found that the cells that showed the most depression for 0 showed very little at the other intervals, because now she's doing short-term depression. She can test each cell on all of the different pairing intervals. The ones that liked 0 didn't like the other intervals. The ones that liked 100 didn't like the other intervals, and so on. So although if you just average them all together, it looks like there's not a temporal specificity, but this seems to be coming from averaging together different cells that are tightly tuned for different intervals, rather than a lack of tuning in the cells. And so to quantify this, we used a selectivity index. So that's just how much depression you get at the best interval versus the others. If you do this on the population means, the vermis doesn't look selective, whoops, although the flocculus does. But if you do it on individual cells, then the individual cells are almost equally, non-significantly different from the flocculus in terms of how tightly tuned they are. So now getting to your question, Peter, about function, we were able to find, so the punchline is different parallel fiber to Purkinje cell synapses are tuned for different delays. We were able to discover this by working on the flocculus, where all of the cells are contributing to eye movements. All of the climbing fibers carry visual signals with a common delay. Even though we restricted in the vermis our experiments to 2 lobules, there's a lot of functional heterogeneity in those lobules. And we know that those 2 parts of the vermis, some of the climbing fibers are coming from the spinal cord, carrying somatosensory information at very short latencies, tens of milliseconds. But these lobules also light up in imaging tasks, for cognitive tasks, where the latencies would presumably be longer. So what we think is that there's different cells are looking for different intervals, and we would hypothesize that that maps onto the functionally relevant delay for the error signals coming into each cell.
Question. So you're saying within one Purkinje cell, you don't have heterogeneous synapses that like different delays?
That's a prediction that's
That will
testable.
not find
Yeah,
that.
yeah, because each cell gets input from one climbing fiber, and the delay between that Purkinje cell's activity and the feedback coming in on its climbing fiber input should determine the preferred interval for all of its
Then
synapses.
if you move one Purkinje cell over, do you expect the delay to be different, or do you expect in a region to have a regional delay?
Yeah, we looked for mapping within the vermis and couldn't pull it out by comparing across slices, but that's something to go back to look for more clustering on a finer scale with a better technique. So let me just summarize the same story that you heard from Jim, that the cerebellum has been described as crystalline in its architecture, but within that crystalline architecture, there seems to be important functional heterogeneity that matters. We shouldn't think of these brain areas like cerebellum and basal ganglia and hippocampus as monolithic, but actually having a particular architecture on which is layered important heterogeneity. And then by looking at the flocculus, we were able to see that that heterogeneity reflects a functional tuning for specific task demands. So I'll stop there, and I saw Dan had his hand up.
That short-term depression is thought of as a presynaptic phenomenon usually, and if so, is the specificity for those temporal windows related to the identity of the parallel fiber or to the identity of the Purkinje cell that wasn't clear to me?
Yeah, so the long-term depression at these synapses is postsynaptic, and I'm pretty sure we looked for a change in paired pulse ratio with the short-term depression and did not see it.
I guess my second question is, are you imagining that the time scale of the long-term plasticity was somehow preset genetically and then learning scaffolds on top of that, or was it another plasticity that set that learning window to the right window?
Yeah. So this observation, this finding of the tuning, I think raises many interesting questions. How do you generate it at a cellular level? But this question that you're asking is our favorite that we're working on right now. Did this just arise through evolution and then the right circuits learn to take advantage of that heterogeneity? Or is it actually tuned by the statistics of delays that it sees? And we're testing that by perturbing the normal delays. And we don't quite have the answer yet, but it's going to be interesting. Either way, it's going to be interesting, I think. Lauren.
Yeah, so this is sort of a follow-up. So I'm struck by this, and we talked a little bit yesterday about results like from hippocampus, where there's now like a 2-second window where you can potentiate things that are at least a few seconds in the past, which really suggests that there's a whole new toolbox of things that people could use to learn almost any temporal relationship they want to. And I just like this suggests things like synaptic tagging and weird biochemical time courses. So I'm just curious, do you have any guesses, or is there anything out there that makes you think, okay, this is sort of the kinds of mechanisms we'd expect to see? that would support it? You know,
Oh, you mean what are the cell, how do you build
how do
a
you
narrow
actually
window?
do that?
Yeah, at first I thought the hard thing is how do you get such a narrow window at such a long delay? But as I thought about it more, all you need is something analogous to the action potential, where you have something that builds up to a threshold that opens the window for plasticity and also triggers its close. And so then the question becomes, how do you build up to that threshold with such a consistent time course and with a different time course in different Purkinje cells? And so that's still a hard problem. And every cellular physiologist I talk to has a different pet idea. You know, it could, Serena Dudek works on calcium pumps, so she thinks it's a calcium pump. So it's going to take an unbiased approach to try to get to that, I think, because there are too many candidates. Maybe the question.
You said at the beginning how if someone gets a different prescription for the glasses, there's relearning involved. Now, typically, if someone has trifocals, you've got 3 different prescriptions. So would you have 3 different control channels, or would you have almost instantaneous learning in one control channel?
Yeah, so there's a whole interesting theory about why half of the neurons in the mammalian brain are granule cells, cerebellar granule cells. And one of the most interesting ideas is that it's so that you can do context-specific learning, so that you can use the context of frames on my face to cue a different VOR gain than in a different context. And context-specific learning is the kind of thing you need for motor precision.
This is really fascinating, but I'm just trying to picture, you know, a Purkinje cell listens to this beam of parallel fiber axons. And so either, you know, if you can assign one time delay to this whole Purkinje cell, which has 100,000 inputs, it suggests that they all have to have the same kind of, you know, the error that they might be, or that they all have to kind of agree on this delay. But then it kind of throws out the window the idea that these Purkinje cells integrate tons and tons of inputs that might come from many, many different places, which have a different responsibility for the delay.
Yeah, so the delay that matters is not like, you know, they're getting auditory inputs that are fast and visual inputs that are slow. The delay that matters is synapses that made the Purkinje cell fire at this time, and the time it takes for that Purkinje cell, whatever made it fire at this time, led to an eye movement that then maybe caused retinal slip, and that gets reported back on the climbing fiber. So it's the time between the Purkinje cell firing, or you know, the synapses making the Purkinje cell fire, and the evaluation of whether that was a good outcome or not. So the time it takes for the parallel fibers to get activated is not relevant. It's the time from Purkinje cell getting activated by the synapses to the feedback coming back.
Thanks very much.